Okay, so we had a lively discussion. Now, let me start off by thanking Bing for really taking the lead on this, and I was trying to help along. We also had a lot of great participation, a lot of great comments. And so functional annotation of the human genome, the main questions we had is what's needed to identify all the regulatory elements, as well as all of the genes in genomes, in the human genome. Second, how can we characterize the function of all these genes and regulatory elements in different biological contexts? And then what specific projects should we suggest that NHGRI try to support? And we had quite a process. We had a list of where we are now. We had and went through where we wanted to get to, and we went through a lot of discussion of how to get there. I will not go through that in any detail, but shift right to our summary. And these are the projects that we that came up that we should consider recommending. And as you can see, they're in the two categories that we've been asked to focus on, the ones that will be ready in the next few years and those that are designed for the longer term. And we actually paused to try to get an assessment of how much buy-in there really was amongst the many participants in the room, and that's what the color coding is. We had a pretty strong agreement to the ones in red, and we either just didn't have time to get a vote or just other things for these ones in black. Okay, so what are we recommending? The first one is a comprehensive atlas of the cis elements in DNA and in RNA. Complete the atlas. The second one gets into this. Okay, everybody wants to know what the elements are, and there's this huge diversity of ideas about how to ascertain whether or not the candidates you've come up with are really active. And the activity, every assay is actually looking for a particular activity, and we lump it all under function. And that's a very broad term. So one proposal, this actually came from John Stamatoianopoulos, that is a way to make some concrete progress in this difficult issue of assessing the function of each of these elements in the catalog, was a pilot project. And the aim is to thoroughly assess function in many different ways, okay? And so this project would choose an appropriate subset of the human genome and choose it wisely to try to cover different phenotypes and other things, and then do the exhaustive perturbation and define those functional consequences, and doing it in maybe cell lines or in tissues, but also really embrace the power of model organisms. Look at the orthologous or even analogous regions in the model organisms. And that was given a initial thing of fun code, but others are already getting pushback for that. But anyway, the idea is a pilot project to help us to come to grips with the complexity of the complexity and diversity of functional assays, and what does it really mean to have sufficient data to make a strong call that this really is a bona fide regulatory element or other element. Okay, another project. This is building off the power of the yeast knockout project, and it was suggested, well, you could take like the haploid, HAP1 cells and do knockouts of all genes. And in fact, it was pointed out, well, that's already been done, and that there's already been a really informative assay of do the cells survive, and maybe more phenotyping of those. So that's a potential project. And I think that this fun code for model organisms, I think, is captured in that second item. If it's not, we'll open up the floor for clarification from the participants. We also had a proposal for a 1,000 epigenome project. That came up, it wasn't discussed a whole lot, but I think that means 1,000 different cell types, although we were debating, well, maybe 1,000 different individuals. and I'm hearing from Eric Mendenhall, it was 1,000 individuals. Okay, as you expand out to different species, the actual gene nomenclature is not harmonized across them all, and there's a real need for a universal vertebrate gene nomenclature. Oh, this has come up in multiple contexts already, but there's tremendous power in evolutionary analyses, and you certainly could come up with an atlas of elements that are under purifying or under positive selection, if you depending on how you do the assessment. There's a lot. This next one, there's a lot of richness in this, and we just covered it very lightly, but as you know, there's a huge amount of information we can learn from the nuclear architecture, and an atlas of that in multiple cell types would be really, really beneficial. Those are short term. Long term. Scalable methods for phenotyping. And hopefully they would learn a lot from this proposed pilot project. Second one, scalable methods to perturb DNA in human and in model organisms. And we've heard some, even just how to even introduce, say, CRISPR reagents into some of these various systems needs to be solved. Here's a favorite one that comes up over and over, and I'm glad to embrace it. End-to-end complete human genome. And there's been, I don't need to say any more right now, and others can join in more, but yes, that would be nice to have a complete genome if you're going to try to say your catalogs are complete. Assemble full genome sequence for 1 million species. That was one ambitious proposal. Here's technologically, now you can do lots of single cell assays. There's like coverage, and there are other issues with that, but as a longer term objective, could we get a full epigenome maps in single cells? And then this almost came up as an afterthought, but it's not because it's not really, really, really important. It is really, really, really important. So let me say, last but certainly not least, comprehensive catalog of protein functions. So you could actually, when you see there's a variance in a certain gene, that you would have some idea of what that, the function of that protein. So that's it for my little presentation. And do we have discussion now from that, or
we're going to hold discussion?
Hold discussion? Okay, all for now.
Just see if anything was missed by your co-chair or
there we go.
people in
That's
the group.
good instructions.
Just
Did I miss something?
Just very briefly
Okay.
before we go into discussion.